Am I audible and visible? Let me know, then we'll start. Am I audible and visible? Fine. Hello everyone. I'm myself Dr. Deepti Karya. I'm MD PhD Physiology and your faculty for Physiology. Okay. Now, so today we'll discuss about regulation and you can say phases of gastric secretion. Okay. Before starting my class, I would like to inform you all that you will get all the notes of my classes in soft copy as well as in hard copy. For soft copy, you have to download the application Vesalius and subscribe, and for hard copy you have to order on Flipkart and Amazon. Link for all that is given for the hard copy as well as soft copy, as well as you can join my WhatsApp group. The number that is 78638 22668. Okay, this you can send me message directly. You can ask your any of the query on my WhatsApp group, or give me a message personally. I will try to solve all your queries related with physiology. Fine. Now, today we'll discuss about phases of gastric juice secretion. Okay, so here, as we have discussed, like salivary secretion, gastric juice secretion also is in three phases: cephalic or cephalic phase, gastric phase, and intestinal phase. So physiological regulation of gastric juice, that is in three phases: cephalic or cephalic phase, gastric phase, and intestinal phase. Okay, you can see here, this is cephalic phase by vagus nerve, gastric phase that is by nervous reflexes, vagal reflex, as well as hormonal stimulation, and intestinal phase that is by neural and hormonal mechanism. We will discuss in detail. Starting with first one, that is cephalic or cephalic phase. Cephalic or cephalic phase, that means gastric secretion takes place before the food enters in the mouth. Okay, here you can see either psychic stimulation by thought of the food, by smell of the food, by you can say hearing about the food, that increases the secretion. It is same way as we have discussed in the salivary secretion. This is known as appetite juice, because here food is not there in the mouth, but still salivary secretion as well as gastric secretion increases. And what is the cause here? Because we are knowing the taste of the food previously, so reflexly, because we are knowing the taste, the food, that test which is conditioned, that is a conditioned reflex, either sight of the food, smell of the food, or thought of the food, or vision of the food. If we see some favorite food, we have salivary secretion. Same way we have gastric secretion. So this is known as conditioned reflex. This is not inborn, that is not available since birth. Means, suppose if you put good food in front of newborn baby, then in that baby there is no salivary secretion, no gastric secretion, because the baby doesn't know the taste of the food. Because we know the taste of the food, we have increased salivary and gastric secretion. We don't have gastric and salivary secretion for bitter food. So this is always a conditioned reflex, and this is vagally mediated. Now, so the pathway for the same, that is, you can see that neurogenic signals for this cephalic phase, they are originated in the cerebral cortex, mainly in the appetite centers of the hypothalamus and amygdala. Hypothalamus and amygdala. Okay, and these impulses are transmitted to the dorsal nucleus of vagus nerve that is present in the medulla. Okay, and from which efferent fibers, they travel, they are transmitted in the form of the vagus nerve, and that reaches to the stomach, and that increases salivary secretion. It increases secretion from chief cells. So pepsin or pepsinogen secretion is increased. Also it increases secretion of gastrin from G cells. Okay, and also it increases secretion of HCl from parietal cells. So this is the secretion which is rich in pepsin, gastrin, and HCl. Fine. Now, what is the rate of gastric juice secretion by this cephalic phase? This rate is highest. You can say that this is 500 ml per hour, but the phase is not lasting longer. Okay, so it is responsible for only 45% of total secretion. 45% of total gastric secretion, that is by cephalic phase. Okay, this is a vagally mediated gastric secretion, so it is affected by emotions because it is concerned with the cerebral cortex, hypothalamus and amygdala. So you can say that if there is anger, uh, emotions and hostility, gastric acid secretion increases. You might have felt that if you have anger, if you have hostility, you feel gastritis because of the increase in the gastric secretion. Motility is also increased, and fear and depression decreases gastric secretion and motility. And this is the reason, because it is vagally mediated and influenced by higher centers. Okay, second phase, that is gastric phase. Here food is there in the stomach, okay, and that increases gastric secretion. Now how this increases gastric secretion? That is by one, that is local reflex pathway. Okay, this reflex arc that is there in the wall of the stomach, as we have discussed, that mesenteric and mesenous plexus and all. So first thing, that is, you can see here, when food is there in the stomach, there is distension, stretching of the stomach wall. So you can say that there is a mechanical stimulus that initiate local reflex. Second thing is chemical stimulus. Chemical stimulus means when the food is there in the stomach and it is digested and it is converted, or you can say it is converted into products of protein. Okay, small amino acids are formed. So this chemicals also stimulate the local reflex pathway. So that is the first way. Now this fibers from these receptors, either mechanical or chemical stimulus, that enters in the mesenous plexus. We have discussed mesenous plexus is also known as submucosal plexus, that is present in the submucosal layer. Okay, so this mesenous plexus, that fibers, they reach to the mesenous plexus, and they synapse on the postganglionic parasympathetic fibers, and this postganglionic parasympathetic fibers, they end on the parietal cells, and they increase the secretion of HCl. So gastric acid secretion increases. This is the first way, local reflex pathway. Second, that is by the product of protein digestion. When the product of protein digestions are there, they increase. This is hormonal way. You can see here, this is your stretch receptor. So this is one. Second is hormone, that is gastrin. Product of protein digestion, they increase gastrin secretion, and this gastrin secretion, gastrin in turn increases secretion of gastric acid. Okay, so that is second way. Third way, that is when the pH is low, there is increase in the pepsinogen secretion through local pathway, because at low pH or high HCl, H plus concentration, this pepsinogen is converted into pepsin, and more amount of proteins are digested, so more amount of amino acids are formed. Okay, next is rate of gastric juice secretion. Here this is less than cephalic phase. Here this is 200 ml per minute, but this phase lasts for longer time. So total amount of gastric secretion in this phase is 50% of the total. Okay, fine. Now, next is intestinal phase. When food enters in the intestine, here, presence of the food in the intestine, here, there is inhibition of gastric secretion. Okay, it has got influence on the gastric secretion. How? Because now these fats and carbohydrates and proteins, they all are in the duodenum. So there is release of certain your hormones and enzymes. This gastric inhibitory peptide, secretin and other GI hormones, they inhibit gastric secretion, because now food is not there in the stomach, so gastric secretion is not required. And it is proven that when this intestine is removed, there was a practical there was an experiment which was done, that when intestine is removed, what happened, this gastric secretion increases. So you can say that these are the factors, they inhibit gastric secretion. Okay, now another reflex, that is enterogastric reflex. Enterogastric reflex, that is when food is there in the small intestine. Okay, this enterogastric reflex is initiated by distension of the small intestine. Either there is presence of acid, or there is presence of the degradation product of the protein in the upper small intestine, and this intestinal mucosa gets irritated. That reflex is generated, and this reflex activity, it is passed through intrinsic as well as extrinsic pathway. Okay, and it stimulates sympathetic and vagus nerve, and mainly sympathetic activity is dominated, and there is decrease in the gastric secretion. Okay, another is hormonal mechanism also is it plays an important role because of presence of acid, fat, and hyper or hypotonic solution in this duodenum, upper small intestine, that releases various hormones, as we have already discussed: secretin, cholecystokinin, gastric inhibitory peptide, vasoactive intestinal peptide, polypeptides, and vasoactive intestinal peptide, and somatostatin, and they all also inhibit gastric secretion. This is hormonal mechanism. Okay. Now another is product of protein digestion. This product of protein digestion, they stimulate gastric acid secretion, and this is 50%. This accounts for 50%, or you can say total gastric secretion, 5% of total gastric secretion, that is after meal, because of this products of protein digestion. Okay, so again we are repeating these three phases. Cephalic phase, when the food is not there in the mouth. Okay, vagus mediated. Okay, and here because of the vagus mediated, there is release of acetylcholine, stimulates parietal cells and G cells. Gastrin mediated, hormonal regulation, that also increases gastric acid secretion. In gastric phase, there is local reflex. Okay, local reflexes, they also stimulate. There is distension of the stomach, also stimulates gastric secretion. There is hormone gastrin from G cells, also stimulate the gastric secretion. And intestinal phase, when products of protein digestion are present in the intestine, the gastric secretion decreases. Okay, here hormones for inhibition of gastric secretion, they are GIP, secretin, cholecystokinin, vasoactive intestinal peptide. Fine. Now, what are the other influences they affect gastric secretion? One, that is hypoglycemia, when blood sugar level decreases, gastric juice secretion increases, both acid secretion as well as pepsin secretion. And this action is through brain and vagal fibers. Okay, vagal efferents. Okay, another is when person takes alcohol and caffeine, that also directly acts on the gastric mucosa, and when it acts on the gastric mucosa, it increases acid pepsin secretion. And emotional factors, they also affect. We have already discussed anger and hostility increases gastric secretion, and fear and depression decreases gastric acid secretion. Fine. So these are the other influences. Now very important point comes here, that is experimental demonstration of the regulation of gastric secretion. That is first, that is shame feeding. Shame feeding here, by these experiments, what we do is we are finding different phases and how the gastric acid secretion is regulated. So in shame feeding means false feeding. We are feeding the animal, but this food will not enter into the stomach. This has been removed outside. So how that is, how this experimental animal is created for this experiment. Here, by this experiment, we can demonstrate the cephalic phase, how we will discuss cephalic phase of gastric secretion. For that, what is the procedure? Here, esophagus of the animal, that is to be cut, and it is exposed, and it is divided in the middle of the neck, and these two cut ends are placed to the surface and they are stitched. So whatever food the animal is taking, that will not go into the stomach, that will be removed here. Okay, so the food is there in the mouth, it has not reached to the stomach. Okay, but still there is gastric acid secretion. So when the animal swallows food, the food comes out through the upper cut end of the esophagus, it is not going in the stomach, but still there is gastric secretion. Okay, as well as the gastric secretion is here what we can do is we just have to place some cannula in the stomach, and we are collecting the gastric juice to find out that how much gastric secretion is there. Okay, so you can say that after sight, smell, or taste of food, okay, there is gastric secretion. So this proves that there is, you can say that, cephalic phase of gastric secretion. Okay, another important thing here is the gastric juice we are collecting here. This shows certain characteristic. Number one, that is appetite juice or cephalic phase gastric juice secretion. It starts after 5 to 7 minutes of the food there in the mouth. Okay, that is one thing. And the volume reaches to the peak within 1 hour, and secretion continues for 3 hours. And this gastric juice is rich in pepsin and it is also rich in acids. Okay, and so same type of gastric juice secretion is produced when we are producing hypoglycemia by injection of insulin. So you can say that when we are producing hypoglycemia by injection of insulin, we are stimulating vagus nerve. And here also in cephalic phase, we are stimulating vagus nerve. So this proves that vagus is a secretomotor nerve. So here another experiment is also performed, that is Pavlov's pouch experiment, that is to demonstrate that vagus is a secretomotor nerve to the stomach. That we will also discuss how this Pavlov's pouch is produced. Okay, so for the formation of Pavlov's pouch, what we do is, under general anesthesia, there is a small pouch which is created from the stomach. You can see here. Okay, and what is the characteristic of Pavlov's pouch? Here the pouch has intact nerve supply and blood supply. Along with that, one more experiment is to be performed, that is vagus is exposed and divided in the neck, and vagus is stimulated to find out whether when we are stimulating vagus we will get gastric secretion or not. Okay, now the experiment, you can see after some days, when this whole procedure, peripheral ends of the vagus nerve is stimulated in the dog which is unanesthetized. So when we are stimulating vagus now, what happens? There is secretion of gastric juice which is rich in HCl and pepsin. So you can say that vagus is a secretomotor nerve. That is one thing. Another kind of pouch that is produced, that is Heidenhain pouch. Here what we do is, this is same type of pouch like Pavlov's pouch. The difference is here this is quite modified type of Pavlov's pouch. Here what they do is, nerve supply is also cut, denervated pouch. Blood supply is intact. There is a pouch which is created which has intact blood supply but no nerve supply. So you can say that when the food is there in the stomach, in distension of this denervated pouch, along with secretion of gastric juice, here there is also secretion of gastric juice. Okay, as well as you can say that when we are injecting gastrin hormone, then also there is secretion of gastric juice. Okay, so you can say that there is interaction between nervous as well as humoral mechanism. So in sham feeding, you can say that there is marked reduction in the secretion from parietal cell if gastric secreting pyloric enterum is removed. But when we are infusing gastrin, this secretion increases. So you can say that nervous factor, that is by vagus nerve, that increases gastric secretion. And if we are cutting this nerve supply like in Heidenhain pouch, what happens? Still there is gastric secretion, that is because of presence of the hormonal influence, that is gastrin. Okay. Other types of denervated pouches are, one is Bickel's pouch. In Bickel's pouch, it is similar to Heidenhain pouch, but blood supply is also to be cut and sympathetic nerves also to be removed. But still here there is gastric acid secretion if we are stimulating the main pouch of the stomach. Other is Farrell and Ivy's pouch. Here what they do is, they produce a pouch, they separate the pouch and they keep the pouch in the abdominal cavity somewhere else. But still here also gastric acid secretion is there. So you can say that there is one hormone, gastrin, which passes through blood and it increases secretion. Okay, so these are different experiments. They are performed to find out secretion of gastric juice during different phases of gastric secretion, cephalic phase as well as gastric phase and intestinal phase, as well as we can come to know the effect of vagus nerve and we can come to know the effect of gastric. Okay, so this is all about phases of gastric secretion. Okay, I repeat again, if you wish to get soft copies of all my lecture topics, it is available on the application Vesalius. You can subscribe and you can download and subscribe this application and you will get all the soft copies, and for hard copies you can order on Flipkart and Amazon. That is available. My notes are available on Flipkart and Amazon. Link for all that is given in the description box. You can also join my WhatsApp group. The number is 78638226668 and you can WhatsApp me regarding any of your query. I will try to solve your query of physiology as soon as possible. Okay, thank you so much. If you like my video, you can like it, you can share with all your friends and subscribe my